afternoon. This is laboratory exercise 12, lab video, about skeletal muscle and the sarcomere. So to start off with, what we are looking at is in exercise 12, figures 12.1 and 12.2. And just to review, please remember some of the important terminology. When we talk about muscles, we're talking about the organ. So for example, the biceps muscle is the entire organ. That is made of bundles of cells called fascicles. The entire organ is surrounded by connective tissue called epimysium, and those bundles or fascicles are surrounded by tissue called perimysium. Within each of those bundles, we have the individual muscle cells. Those individual muscle cells are surrounded by endomysium. Now, if you take a look at the entirety of the muscle cell, within the muscle cell, we have an arrangement of these very specific organelles that are called myofibrils. And the myofibrils are made of repeating units called sarcomeres. When we look at contraction in skeletal muscle, it's actually an effect of contraction within these functional units called sarcomeres where the contraction occurs. So that's what we're going to look at using a sarcomere model. So this is a three-dimensional representation of a sarcomere. What we have represented here is one of the repeating units that you would see within the myofibril. Now in terms of what we're seeing here, there's this area that is green. Okay, and notice there's a little glare over here on this side. What we're seeing that looks like this green webbing, this is a special organelle. It's a type of endoplasmic reticulum or smooth endoplasmic reticulum, specifically called the sarcoplasmic reticulum. These yellow lines here, these represent T-tubules or transverse tubules. If we were looking at the entirety of a skeletal muscle cell or a muscle fiber, we would see many of these sarcomeres in a row creating the myofibrils and many myofibrils stacked. And this is reflected on the diagram from the lab book in figure 12.2. So to just quickly review what happens in terms of contraction in the skeletal muscle, all skeletal muscles need to receive innervation from a neuron. So a neuron sends an action potential down to the neuromuscular junction. If you want to review neuromuscular junction, there is a separate lab video that goes over neuromuscular junction. At the neuromuscular junction, the neuron releases the neurotransmitter acetylcholine. That acetylcholine binds to receptors on the motor end plate of the muscle cell, leading to the opening of sodium channels and depolarization. If that depolarization is substantial enough, it will lead to an action potential within the muscle cell. Once the muscle cell has undergone an action potential, that action potential will travel along the sarcolemma, or the plasma membrane of the muscle cell, and it will pass down through all of the T-tubules. When that action potential passes down through these T-tubules, it will induce opening of voltage-sensitive calcium channels. Those will open, and stored in here within the cisternae of the sarcoplasmic reticulum, we have calcium ion. That calcium ion, when those voltage-sensitive calcium channels open, is going to leave the sarcoplasmic reticulum and is going to move to the sarcomere. So to see what happens in the sarcomere, I'm going to remove this cover. So basically I'm removing the T-tubules in the sarcoplasmic reticulum. Now when we take a look at the sarcomere, this is the smallest functional unit of contraction that we have in the muscle. We have a few different important structures here. This center line here, this is called the M line. Right? Think of this as the middle. This is where contraction will move to. These red areas here, these are thin filaments. Thin myofilaments are made of the protein actin, as well as the regulatory subunits of troponin and tropomyosin. These blue and black lines here, these are the thick filaments, and those thick filaments are made of the protein myosin. We also have, at the edges, we have what's called the Z-disc, and the Z-disc is where you would have one sarcomere abutting the next sarcomere. Shown here is just one sarcomere. So the way contraction occurs, looking at it in this model, when we have calcium released from the sarcoplasmic reticulum, the calcium makes its way to the sarcomeres where it binds on the troponin regulatory subunit. That troponin changes shape and it pulls back the tropomyosin that is blocking the actin binding sites that will allow myosin and actin to interact. When we have actin binding sites available for myosin interaction, the myosin heads will hook in through what's called the cross-bridge cycle, and will then ratchet forward using ATP for energy. What that leads to is it leads to the movement of these thin filaments towards the M line, and you can see how these would move together. This shortening or contraction of a single sarcomere is a very, very tiny movement. However, when these sarcomeres are repeated throughout the myofibril within the muscle cell, we see that we would have a large amount of contraction over the course of each sarcomere contracting the whole myofibril. All of the myofibrils acting in a coordinated fashion would contract the muscle fiber, or the muscle cell. When all the muscle cells or fibers contract, we get contraction of the fascicle, and when all the fascicles contract, we then have contraction of the entire muscle. So just to see then how this relaxation would occur when calcium is taken back up into the sarcoplasmic reticulum because there is no longer acetylcholine binding at the motor end plate. The actin binding site would get covered up again, myosin would disassociate, and these would go back to their resting position. So once again, the way we see contraction in the sarcomere is we have the calcium binding, myosin interacts with actin, brings the thin filaments towards the M line, moving relative to the thick filaments, and relaxation would occur once calcium is removed. Now, if you'd like to see an additional video for this, there is a 3D animation in the A&P Flicks that shows this very nicely as well. Good afternoon. This is the review video for the neuron and the neuromuscular junction, coming from exercise 15 and also part of exercise 12. So to take a quick look at this, I'm going to show you this diagram, or this model. This model shows a multipolar motor neuron, and it's in a diorama that allows you to see how this multipolar motor neuron is going to interact with muscle in a neuromuscular junction. So let's start off looking at the portion of the actual neuron. So this would be the body or the soma of the neuron. These regions here, which are cut off in this three-dimensional model but can be seen over here, those are the dendrites. The dendrites are the receptive regions, and they receive signals coming in from the axonal terminals shown here and here and here from neurons previous in the sequence. At the axonal terminals of one neuron going to the dendrites of another neuron, we see chemical exchange of information through neurotransmitters. Signals are sent from these dendrites in the forms of graded potentials, and if the graded potentials are large enough here within the cell body, they will summate, and in this area here known as the axon hillock, we will see these graded potentials being converted into an action potential. For a review of action potential, watch the A&P Flicks videos on mastering A&P. Once an action potential has begun, that action potential is going to proceed down the length of the axon. Now in motor neurons, those axons are myelinated, so they have these regions called myelin sheath, which are made from Schwann cells. And if we remove one section here, you can see that we have here is actually a layering of the plasma membrane of a Schwann cell, and it creates basically an insulation. This allows for the speeding up of signals that are being sent in terms of action potential. When this action potential comes down, it goes down the entire length of the neuron. Neurons are bundled into larger groups called nerves. Those nerves would terminate, depending on the type of nerve. If we were talking about a motor nerve, would terminate on muscle. Okay? So this part over here depicts a muscle and what's called a neuromuscular junction. A neuromuscular junction is where you have the axonal terminal of a motor neuron in communication with a muscle cell at what's called the motor end plate. And if you look at the previous video on sarcomere functioning, what occurs at the neuromuscular junction is the motor neuron, when the action potential reaches the axon terminal, it induces the opening of calcium channels. Calcium influx is then going to induce the movement of synaptic vesicles full of neurotransmitter called acetylcholine. That acetylcholine is going to be released through exocytosis from the axonal terminal of the motor neuron. It will then cross the synaptic cleft, binding with acetylcholine receptors on the muscle, inducing an action potential in the muscle cell. That would then lead to contraction. If you have further questions or concerns about action potential, use the Mastering A&P Flicks to take a look at how action potential and graded potentials occur.